for the end term exam. So we want to just quickly move through it. Want to go through it very fast so that at least we are at par. Now, the exam you did was not difficult as such, but majority of you is like you are not prepared very well for this paper. This was one of the easiest paper 1 exam you did. So basically, when you look at the questions, they were very familiar, but the problem with this is that sometimes you tend to move very fast and you don't look at these questions very keenly before you start answering them. You just throw your answers aimlessly. So I'm very sure by the time we are done revising this paper, if you are given similar paper, of course not similar questions, I'm very sure you will be able to give us a better performance or better results than the one you gave us last term. Now let me hope all of you have questions with you. You have the papers with you, so I expect you to have a pen and at least somewhere to write. So if there's anything you need to add, of course, feel free to add on the question paper. And if you have a shorthand notebook, of course, you can also have a summary on the same. Now, question 1, explain the following. Explain the following. Of course, question 1 came from which topic? Transport, of course, in plants. So when transplanting, of course, on the question paper, it was mistyped. It was not type 12. It was typed transporting, but supposed to be when transplanting a seedling, it is advisable to remove some of the leaves. So why do we do that? Of course, to reduce rate of transpiration. It reduces rate of water loss through transpiration and also prevents wilting. So again, you need to understand what wilting is. And of course, wilting is a process whereby plants, the rate at which plants lose water the rate is faster than the rate at which the roots are absorbing water from the soil. So of course, we are reducing rate of transpiration and also reducing wilting. Part 2 of the question 1, was there are generally fewer stomata on the upper side of the leaf than on the lower side of the leaf. So still this is on the same topic, transport in plants, and the same answer applies here. It reduces rate of transpiration. Reduces the rate of transpiration. Question number 2. Question number 2. Of course, still in transport, but this time around, transport in animals. We are looking at the function of mammalian blood other than transport. Functions of mammalian blood other than transport. 1 is protection or rather defense against infections or pathogens. Number 2, regulation of body temperature. We also have regulation of pH, and then of course clotting. So you don't talk about transportation here. Here you talk about just other thing rather than transport. So here you are supposed to look at the question very well, read it through severally, and then understand exactly what the examiner is looking for. Question number 3, part A. State 2 antigen types in the blood which are responsible for determination of the blood groups of a person. Of course, this we've been looking at. We know we have antigen A, which is responsible for blood group A. We have antigen B, which is responsible for blood group B. Then we have antigen D, which is also called rhesus antigen. So an individual with antigen A is said to have blood group A, an individual with antigen B, and remember antigens are normally denoted by a capital letter. So an individual with antigen capital B is said to be a person of blood group B. An individual with blood group A, of course, we expect antigen A. So antigens are normally denoted by capital letters, and then antibodies are denoted by small letters. So antigen D can also be referred to as rhesus antigen. Question number 4. Name the structures for gaseous exchange in Roman 1, a mosquito. So when you look at a mosquito, of course, gaseous exchange structure. For this case, we wanted you to give us the word siphon. Some of you are telling us, talking about tracheal system. Remember, mosquito larvae lives in water. So we have air tubes or siphons, or even spiracles. So the answer that was being marked right was the siphons. Look at amoeba. For the amoeba, the structure is a cell membrane. Cell membrane, and the process most of the time is normally diffusion, so it's cell membrane. Fish, the respiratory surface for a fish is not just a gill, of course, to make a gill, but it's supposed to be gill filaments. Gill filament, these are respiratory surface. Gill filament, not just gills. Okay? Gill filament. Oh, these are, we are supposed to look at the structure. Actually, the structure is gills. The structure, gills. So those who just write gill, you are not giving it right, because the question itself is talking about plural form of the answers. So it's supposed to be gill. But if you are asked to give the respiratory surface, then you talk about gill filaments. Talk about gill filaments. Question number 5. State 3 characteristics of respiratory surfaces in animals. 1, they are normally moist, of course, to dissolve respiratory gases. 2, they have thin epithelium to reduce diffusion distance. Then another point, they are highly vascularized, or you can talk of richly supplied with blood vessels. They are highly vascularized, of course, to create a steep concentration gradient for faster diffusion of gases, of respiratory gases, of course. Then we look at some are numerous. If you look at the alveoli, they are numerous. Even the gill filaments, they are numerous, of course, to increase surface area for gaseous exchange. Question number 6. Explain why it is advantageous to breathe through the nose. So one thing you need to understand is that the air is normally warmed when you breathe through the nose rather than through the mouth. Then the same air is moistened, is not dry. You breathe through the mouth, you discover that the air that is coming in is a bit drier. Now again, through the nose, the air of dust. We normally have some hairs in the nose that trap dust particles. So air is normally filtered in the nose before it reaches the lungs. So one, particles are normally trapped, that is filtration. We are talking about the air is warmed, and also there's aspect of the smell of the air is detected, or the quality of the air is detected when you breathe through the nostrils. Question number 7. People living in high altitudes have a larger count of red blood cells. They have larger count of red blood cells. Explain. So one, we say this, at a higher altitude, there is low oxygen concentration. At higher altitudes, there is low oxygen concentration. Therefore, there is need for more red blood cells to increase. There is a need for more red blood cells, of course, to increase oxygen supply to tissues. So we need more oxygen, but what is provided is less at high altitude. Therefore, you need to have more red blood cells so that you can increase the amount of oxygen that is supplied to tissues. Supplied to tissues. We want to look at the meaning of the following terms, because we have respiratory quotient, we have oxygen depth, then we have basal metabolic rate. So the first one, respiratory quotient, or RQ, of course, in an exam, you have to write the formula. In an exam, you have to write the formula. Whenever you're asked to calculate, of course, you write the formula in full, not RQ. Just write it in full, a respiratory quotient, but not RQ. So respiratory quotient is basically the ratio of carbon dioxide produced to oxygen consumed during respiration. You can write it as carbon dioxide produced over oxygen consumed, or the ratio. Whenever we talk about the ratio, of course, is like we are dividing. Sawa sawa. So it's the ratio of carbon dioxide produced against oxygen consumed. What about oxygen depth? Oxygen depth, on the other hand, is the extra oxygen needed after an exercise, of course, to oxidize accumulated lactic acid. In simple terms, it is the amount of oxygen required, the extra amount of oxygen required to get rid of lactic acid accumulated in the muscles, of course, when oxygen supply is lower than the demand. So when the supply of oxygen is lower than what is required, then you experience what is referred to as oxygen depth. So here, some of the students were just writing the amount of oxygen required to get rid of lactic acid accumulated in the muscles when oxygen supply is lower than the demand. Such like students were not able to score, we were looking at the word extra, additional amount of oxygen, not just oxygen, but that additional, the one that is on top of the normal one. That is the one that will get rid of the lactic acid that has accumulated in the muscles. So you're supposed to underline the word extra. Then Roman 3, basal metabolic rate. And this is the minimum energy required to maintain basic life functions when the body is at rest. The keyword here is minimum energy required. And then you're also supposed to look at the word when the body is at rest. So the minimum amount of energy required to maintain the basic life functions or basic body functions when the body is at rest. Question 9 is on excretion in plants and animals, of course. We're looking at functions or economic importance of these excretory substances in plants. So remember, this can be an essay and it can also be a short structured question. So for this case, they have given you these excretory products. 1 is caffeine, 2 is rubber, 3 is tannin, and then there is actually there are 3, but there are more than that. So whenever you're looking at it in form of essay, of course you have to remember all of them, because this can be as an essay and you can be asked to describe economic importance of some of these excretory products. So you have an essay, 1 column, the substance, the 2nd column, the function, the roles or the economic importance, and then the 3rd column, you talk about the source, if you are asked to give the source. So we have caffeine is a mild stimulant. Rubber, of course, when you talk about a mild stimulant, you can talk of is normally used in beverages and medicines as a stimulant. Then we have rubber, which is normally used to manufacture tires, gloves, and elastic materials. Sometimes even making of chewing gums and shoes and so on and so forth. Then we have tannin, which is normally used in the treatment of leather, tanning, and is used as an astringent in medicine. Good. Number 10. Question 10. State 3 methods of excretion in plants. Of course, these are the things I've repeated severally. We are aware of them, but some of you are not able to remember. So that tells us that majority of you are not did not have ample time to revise, did not prepare well for that exam. We have deposition, we have transpiration, we have exudation, guttation, organ fall, and have diffusion. And we have even some other substances that are normally stored in the vacuoles or backs of hardwood or trees. So diffusion, of course, whenever you're talking about diffusion, we're talking about oxygen and carbon dioxide. Whenever you're talking about transpiration, you're talking about loss of water in form of water vapor to the atmosphere. When you talk about we have deposition, of course, those ones we defined. Then we have the other one where water is normally lost, but in form of water droplets. I think that is now the guttation, not in form of water vapor, but in form of water droplets. Number 11. Briefly describe the role of the following parts of the skin in thermoregulation. So thermo, we are talking about temperature. The roles of the skin as far as temperature regulation is concerned. So how does the skin regulate the temperature of the body? That is what we are looking at here. Thermoregulation. So 1, look at the blood. Look at the blood. The blood vessels for this case. So blood vessel can also be stroked with arterioles. So what happens? You have to bring in the aspect of when it is hot, what happens, and when it is cold, what is happened for you to have full score. Now let us begin with the first one. When it is hot, of course, we expect vasodilation of blood vessels. And when it is cold, we expect vasoconstriction of these blood vessels. So when it is hot, they will vasodilate to lose heat to the environment through conduction. And when they vasoconstrict, of course, they reduce heat loss from the body. So vasodilation increases blood flow to the skin, releasing heat. Vasoconstriction conserves heat. Number 2, we are looking at the sweat gland. We are looking at the sweat gland. So in the sweat gland, of course, we are talking about when it is hot, what happens? The sweat gland will secrete more sweat to the skin. The water in the sweat will evaporate, therefore absorbing latent heat of vaporization and then cooling the body. Let me give you just a sec of like 1 minute if I'm too fast so that you are able to write. 1, vasodilation increases the flow of the blood to the skin, releasing heat. Vasoconstriction conserves heat. So you should know when vasoconstriction is taking place and when vasodilation is taking place. Vasodilation when it is too hot, vasoconstriction when it is too cold. On the sweat gland, of course, when it is hot, of course, you expect more sweat to be secreted by the sweat glands. And the water in the sweat is absorbed, or rather, as it evaporates, it absorbs latent heat of vaporization, bringing about cooling effects on the body. Unaona, hii ni inatengenezwa kwa YouTube alafu wanaonea huko, so sio Zoom tu wana respond. Sasa tuko nao watapatiwa baadaye. Nashangaa mbona. So that's why. So of course, on the comment section, if you have a burning question, you can just post it there so that when we shall be having our next lesson, I'm able to respond to them. So that question had 4 marks. So the first 2 marks was on blood vessels and how it regulates body temperature. And the second 2 marks, of course, was on how sweat glands will regulate body temperature. So you can see there's a table here on question 12. The table below shows the approximate concentration of certain substances in the blood plasma, glomerular filtrate, and urine. Study the table and answer questions that follow. We have substances, of course. We have water, proteins, urea, and glucose in the first column. The second column, percentage in the blood plasma. Of course, you can see water is 100%. Protein is 6.5%. Urea is 0.03%. Glucose is 0.1%. 0.1%. Then when you look at the filtrate that is normally formed, in the filtrate, there is 90% water. So you can imagine what is happening. So it tells you that more water will pass through the glomerular. Then we have 0%, of course, protein, so there is no protein. Then we have 0.03 glucose, no urea. Then there is 0.1 glucose in that filtrate. Now, in the urine, water is 60%, 0%, of course, proteins, 1.8% urea, and then 0% glucose. Question part A, why is the concentration of protein in glomerular filtrate and urine 0? So of course the reason why protein is 0 here in the filtrate means that one, protein molecules are too large to pass through the glomerular membrane. So the large size of the protein cannot allow it to pass through the glomerular membrane. Then you look at the urea rises in the urine. Where is it? Not that one first of all. So one, we are talking about the large molecules of the proteins. And then you're also talking about all the proteins are normally reabsorbed at the PCT. All the proteins are normally reabsorbed at the PCT. At the PCT. Explain why there is no glucose in the urine. Of course, all glucose is normally reabsorbed at the proximal convoluted tubule. Because pale juu hakuna PCT. Pale juu tulikuwa tunaongelea tu large sized protein molecule that cannot pass through the glomerular membrane. That was the first thing, 1 mark. But now, the reason why we are not having glucose completely in the urine, look at the glucose. Glucose in the urine is 0%. So the reason why we don't have any glucose in the urine is that all the glucose is normally reabsorbed at the proximal convoluted tubule. Explain the meaning of the following terms. Alteration of generation, of course, we talk about existence of organism in more than one life form or more than one cycle. Existence of an organism in more than one life form. That is, we have the sporophyte and the gametophyte life forms. Or rather, we talk about the life cycles. Life cycles. An existence of an organism in more than life form or life cycles, that is sporophytes, and the sporophyte and the gametophytes. Sporophytic and gametophytic forms. Prokaryotic, cells whose nuclear content is not bound by a nuclear membrane. In simple term, is a cell with no nuclear membrane. A cell with no nuclear membrane. A cell with no nuclear membrane. Of course, eukaryotic is the opposite of prokaryotic cell. So eukaryotic, these are cells that have nuclear content bound by a nuclear membrane. Prokaryotic, their nuclear content are not, or the nuclear inclusions, they are not bound by any nuclear membrane. So we want to look at the second, the question number 13. The aim of this experiment. First of all, look at just the thermometer. The thermometer alone should tell you what is happening. Of course, to investigate production of heat during fermentation or during the anaerobic respiration. So of course, if you look at them, the first one, the thermometer reading is a bit higher, and the second one, the thermometer reading is very low. So it tells you that in the first setup, the setup A, there was anaerobic respiration, and in the setup B, there was no respiration taking place. Therefore, the temperature was constant. So of course, that comes as the second part to account for the observation that has been made after 30 minutes in the setup. So in A, there was rise in temperature due to fermentation. In B, the temperature remained constant. Now, why is it that in B the temperature is remaining constant? Boiling denatured the yeast. Therefore, no fermentation took place. So how do you score the 3 marks here? The first mark is in A, there was rise in temperature due to fermentation. The second mark is in B, the temperature remained constant. And then the third mark is on the reason why setup B, the temperature did not rise. So the reason is because boiling denatured the yeast. Therefore, no fermentation took place. Question number 14. State the characteristics that can separate the following organisms into their respective classes. Characteristics that can separate the following organisms into their respective classes. Now, we have the arthropods. Of course, first of all, we are having millipede, tsetse and a spider. So what are the features you can use, of course, to put them into their respective classes? One, look at the number of appendages or number of legs. Number of legs. So for millipedes, of course, you expect it to have more legs than the tsetse and the spider. So you can easily use the number of legs to classify them. Number 2, look at the presence or absence of wings. Some have wings, some don't have wings. So that one can also be used to separate them or rather classify them. Then we look at the number of antennae. Some have antennae, others don't have antennae. Some have a pair, some have more than one pair of antennae. So that can also be used to classify them. The number of body parts. Have some that are cephalothorax, where the thorax and the abdomen, the head are fused actually. Then we have those that have 3 body parts. So for a tsetse, of course, you expect it to have 3 body parts, and then the spider has 2 body parts, of course, and then the millipede, of course, the head is normally fused on the trunk, that is with the thorax. So that is under classification. Under classification. Question 15. Name the blood vessels that transport blood from one heart to the lungs. Of course, which topic? Talking about transport. So of course, this is very simple. Pulmonary artery. Pulmonary artery. The one that is responsible for transport of blood that is in the small intestine to the liver is hepatic portal vein. Hepatic portal vein. Hepatic portal vein. The liver. Number 16, state the role of light in the process of photosynthesis. So what is the role of the light? The role of the light, of course, is to split water molecule, in bracket, can be photolysis of water, to release energy in form of ATP and oxygen. Now, the dark reaction, of course, the product, we have glucose on the glycerate and so on and so forth. So we have glucose, or in bracket, we have glycerate, 3-phosphate. But the most important thing, what we need to understand, is that water molecules normally split into oxygen, hydrogen, and also ATP. But hydrogen is normally moved to the dark stage. That is carbon 4 oxide fixation. Name products of dark stage. Of course, can have glucose, carbohydrate, fatty acids, amino acids, etc. Give any 2 benefits of blood clotting. Of course, 1, prevent excessive bleeding. 2, prevent entry of pathogens into the body. Prevents entry of pathogen into the body. State the function of the following parts of a light microscope. Of a light microscope. A light microscope. Coarse adjustment knob. Moves the body tube greater distances. Removes the body tube greater distances to bring the image into focus. Not sharp focus. Sharp focus is when we will be talking about the fine adjustment knob. So this one is just to bring the image into focus, not sharp focus. It moves the body tube greater distances, not just great, but greater distances. Or it moves the body tube greatly to bring the image into focus. B is condense, of course. Concentrate light onto the stage. Concentrates light onto the stage. Then, of course, we are having number 19 here. Number 19. Number 19. Number 19. Of course, in number 19, there was a diagram here. We don't have it here on the board, but when you're having, of course, if you have your paper, you can easily see that question. Question number 19. Yes, there's that diagram. Question 19. So use the diagram below to answer questions that follow. Because you can see the diagram, there is glucose under normal circumstances. Then glucose is rising. Then when it is too excess, of course, there's something that is happening so that it is taken back to normal. Then look on the other side, of course, glucose is also going down. And then there is a corrective mechanism that brings it back to normal. Name the feedback mechanism labeled B. So if you look at B, of course, if you look at B, glucose level was high. A hormone Q is introduced that brings it down. So we call it a negative feedback. It is bringing back the excess to normal. Negative feedback. Of course, if you are to be asked to give the name of the mechanism A, because A would be a positive feedback. If, of course, sometimes you can have the excess becoming more excess. That is normally a positive feedback. And the deficient becoming more deficient. Like inaendelea kuteremka kabisa, inaendelea kupanda kabisa. So the positive feedbacks, they are normally trying to make the situation to be even more worse. They worsen the situation. But negative feedbacks, they are normally used to correct the situation. Identify hormone R. Located. We are having this hormone R when the glucose level is lower. So the hormone R is supposed to cause a corrective mechanism, supposed to make the lower glucose in the blood to be normal. Of course, it has to take it up. So the hormone that is responsible for that is normally glucagon. When the blood sugar level goes below the normal, of course, glucagon is normally produced. That, of course, you all know the functions of glucagon and the insulin. So when glucose is low, of course, glucagon will aid in the conversion of stored the stored glycogen into glucose. And when it is too much, of course, insulin is produced so that it aids in the conversion of excess glucose into glycogen, which is normally stored in the body. So hormone R is glucagon. And hormone Q, you are not asked, but hormone Q is insulin. Number 20. A certain animal has incisors, no canine, 6 premolars, 6 molars in the upper jaw, while in the lower jaw there are 6 incisors, 2 canines, 6 premolars, and 6 molars. Write the dental formula. Of course, you just write them as they are. The dental formula, of course, you're supposed to write it as they are. On the upper side, there is 0 incisors, there is 0 canine. So here, you're not supposed to put the word over something. Just put 0 on the upper side. On the lower side, of course, you can see there are 3. Now you are wondering, where are you getting 3 yet there are 6? Of course, at the end of the day, the formula is supposed to be multiplied by 2. So that's why we are dividing everything by 2. So you can see on the upper side, we are having 6 premolars, but we are putting 3. Why? At the end of the day, we will multiply the whole thing by 2. So you have them. Whenever you are writing the formula, you have them. So it is 03, 01, 33, 33 equals to 32. So that is the formula. Identify the mode of nutrition of the organism. Of course, it is a herbivory ama herbivorous. Herbivory mode of nutrition ama herbivorous mode of nutrition. Herbivorous. Some of you are writing herbivores. Herbivores are the animals, is not the mode. So herbivorous or herbivory. Number 22, give reasons for your answer in B above. In B above. Give reason. Why is it herbivory or herbivorous? So there's the aspect of absence of the upper incisors and canine. Majority of you are telling us presence of diastema. You are not able to see the diastema in the formula, but you are seeing absence of some type of teeth. So it's supposed to be absence of upper incisors or canine. But if it was drawn, if it was a diagram that is drawn and indicating there's a part that is the horny pad is visible, then we talk about presence of the horny pad or the diastema. Sawa. Of course, this other one is an ecology. A fish farmer wanted to know the number of fish in the pond. He collected 10 fish in the pond and labeled each by tying a tag on the fins, and then returned the fish to the pond to allow them to mix. When he later collected the sample of 50 fish from the same pond, he found out that 4 of them had a tag label. So when you look at it, of course, this is capture method of estimating population. So first of all, you need to have the formula with you. Is the first capture multiplied by the second capture divided by marked recaptured. So the first capture, we had 10 fish. Then the second capture, he had 50 fish. Now, out of the 50, 4 had some labels on them. So those were the marked recapture. They were captured now for the second time. Therefore, it is 10 multiplied by 10 by 50, I mean, divided by 4, which is equals to 125 fish. Now there's a diagram here. There's a diagram. Now look at the diagram. It shows you a skull of an animal. Name the tooth labeled P and give its function. Of course, the tooth labeled P is a carnassial teeth. Is carnassial teeth. Is the function of that carnassial teeth. The function of the carnassial teeth, sometimes you can even call it canine, is used for tearing and piercing flesh, or slicing through the flesh, and even crushing of bones. So that is the function of P. State the causative agent of malaria and typhoid. Malaria is normally caused by Plasmodium species. You can have Plasmodium vale, Plasmodium falciparum. Then we can even have Plasmodium vivax. But if you don't want to be very specific on which one, just talk about Plasmodium species, SPP. But remember, here, when you're writing them, remember these are scientific names. So you are handwriting them, you're supposed to underline them separately. Some of you are just writing. The answer is correct, but it is not underlined separately. You have not followed the binomial nomenclature system of naming. So you are not able to score at that particular point. So you have to underline them separately. You have to underline them separately. Plasmodium species, of course, P again for this case supposed to be capital, and SPP small letters. For typhoid, of course, Salmonella typhi, again capital S, small t, underlined separately. Question number 25. Question number 25. Define the following terms: carrying capacity. Define the following terms: carrying capacity. So carrying capacity for this case is a maximum population size an environment can sustainably support. Maximum population size an environment can sustainably support, or maximum number of organisms an area can comfortably accommodate without depletion of available resources. Pyramid of numbers. This is the diagram showing the number of organisms at each trophic level in a food chain. The diagram showing the number of organisms at each trophic level in a food chain. In a food chain. So the keyword here is a diagram. Diagram showing, or you can talk about diagrammatic representation. Diagrammatic representation of number of organisms at a particular trophic level in a food chain. Number 26, state one role of a condenser in a microscope. Of course, we had defined that. We had defined that, I mean. Concentrate light onto the stage. Concentrate lights onto the stage. Then explain importance of the following. Importance of the following. Cause one, cutting a very thin section. Cutting a very thin section. So reason why you're cutting a very thick section is to allow light to pass through, or to make light penetrate through it. So allow light to pass through. That is why we normally use a very sharp scalpel, of course, to also prevent, the reason why you're using a sharp scalpel is to prevent distortion of the cells. Using sharp razor to prevent distortion of cells. State two roles of active transport. Roles of active transport. Roles of active transport, reabsorption of sugars and some salts in the kidney. Because we're talking about role of active transport in humans, reabsorption of sugars and some salts in the kidney. Then there's absorption of digested food in the ileum. Then finally, there is excretion of sweat from the body cells. Number 29, of course, explain adaptations of the xerophytes to their habitat. So here you are supposed to use the word some. We've always been singing that, some, not all of them. So you don't say the word, you don't use the word they. You say some. Some have thick cuticle and sunken stomata to reduce water loss. Some have rolled leaves, of course, to trap humid air. Some have deep roots, of course, to access groundwater, and so on and so forth. So we have some have leaves that are needle-like, of course, to reduce surface area for transpiration. Some have more stomata on the lower side of the leaf than the upper side, of course, also to reduce the rate of transpiration. Some show reverse stomatal rhythm, of course, they open during the night and close during the day. Some have succulent stems for storage of water. Then some have extensive root system to absorb underground water, thick waxy cuticle to reflect light, and so on and so forth. Then finally, we had a question on the dichotomous key. There are some leaves there. You can see the first, the second, the third, and the fourth leaf, the fifth one. So with the reason, name the class to which the following leaves are obtained, of course, belong to. N was a class Dicotyledonae. Reason, network venation. Can even talk about being broad. Then R is Monocotyledonae. Reason for that, of course, you can talk about the leaf can be narrow. That can be a reason, and also can talk about the parallel venation. Using the features on the sequence given below, construct the dichotomous key to identify the leaf the specimens above. 1, you are supposed to look at the type of the leaf. 2, you are supposed to look at the venation. 3, you are supposed to look at the leaf margin. And then 4, you are supposed to look at the type of the compound leaf. So we start by saying 1A, based on the first procedure, the first one you're talking about the type of the leaf. So wacha tuangalie type of the leaf. Tunaanzia hapo hivyo tukiongelea A, as far as the type of the leaf is concerned. So there is 1A, leaf simple. We go to 2 because we don't have one leaf, there are more than one. So we'll have to go to 2. B, leaf compound. So whenever you're constructing, remember at that particular point, you just leave it as it is. You don't write anything. You are not directed anywhere because we still don't know where the leaves that are simple will end. So we are going to 2. We are at 2. So at 2, we are at 2. So 2A, as far as the venation is concerned, number 2 hapo hivyo unaangalia unaona pia ni venation. We have leaf network and leaf parallel. So when you look at the leaf network, of course we'll go to 3 because there are also more than 2. We cannot end there. But when you look at the leaf parallel, we only have one leaf that is parallel, and that is leaf R. So tunaandika R. we move to 3. 3 tulikuwa tumeambiwa we look at the margin, leaf margin. So talk about leaf margin smooth as part A, and then part B, leaf margin serrated. So if you look at the leaf that is smooth is Q, and then the leaf that has a serrated margin, look at them, we have N. Then we move to number 4. So remember 4, we are talking about compound leaf. That's why we'll have to go back there. Angalia kwa 1. 1 ndiye imetuleta sasa kwa 4. Yenye tumebaki na hizo compound leaves. Tumemalizana na simple leaves. We are looking at now compound leaves. Then on the compound leaves, of course, we are having leaf pinnate. And then we're having leaf bipinnate. So of course, M is pinnate and then P is bipinnate. Tumemaliza hivyo. So make sure you replay the video so that you are able to answer, to make corrections. So that you are able to make corrections on your paper. So when we shall be meeting next, of course, we are going to look at a topic. So we'll be doing topic called revision. This one was just a revision of the end term. But when we shall be meeting tomorrow, of course, we shall be looking at a particular topic. I'll look at the topic that is normally challenging to you so that we are able to look at it together. But I will also ask for your suggestions on the comment section. Which topic do you think is requires more attention so that we can look at it together? Which topic is more gray to you so that we can look at it together? Otherwise, thank you, and I wish you all the best. Concentrate light onto the stage. Concentrate lights onto the stage.